Hi, this is Michael Edwards, Assistant Professor of Medicine at UC Denver, specializing in bioinformatics. Today's tutorial is Understanding Bioinformatics, Finding the Biology in a List of Genes. What I hope to do with this tutorial is basically give you some understanding or underlying principles behind bioinformatics in a language most people can understand. What I'd also like to do is show you how we can use the bioinformatics to get a better idea of particular genes or pathways or functions that might be important in whatever biological condition that you're looking at. The way I see bioinformatics is more that all of this information we can measure, millions upon billions. I kind of see this as pixels, colored squares, and that each bit alone doesn't mean much. And as you watch TV on your average HD TV screen, you don't see these 2 million little colored squares. What you do is you put this information together to make a cohesive picture. And honestly, that's what I expect bioinformatics to do, and that's kind of how I see its role is. How do we take all these millions upon billions of things we can measure about any given biological condition, say cancer, and figure out how we can use this information to find a potential cure or understand how tumors develop. The easiest way I've found to explain bioinformatics is to use a crime analogy. In this case, whatever you're investigating, say disease or the effect of age on skeletal muscle, will represent as the bank. And it's just been robbed. And as soon as it's been robbed, the call's came out, police put the entire area in lockdown. And given around the bank are all the people, or if we're talking about bioinformatics, genes or proteins that we think might be involved in the bank robbery. And your job as the bioinformatician is the investigator. And at your disposal is the database contained in Scotland Yard. So you have a lot of background information you can use to determine who's guilty or not. So in this case, we would expect most of the guilty parties, if they came in and did a lockdown shortly after it was robbed, to be around the bank. And these I represent in red, and sure enough, these are around the bank. But you'll notice that some of these individuals were just walking by the bank. These are innocent bystanders. And in bioinformatics, we would call these false positives. The key is, how do I find the red ones versus the white ones, the innocent bystanders? It's these red ones that are important, whatever condition we're looking in. How do I isolate those from those that didn't commit the crime? Usually in bioinformatics, we just use simple statistics to do this. This could be a t-test. And this is almost like a picket line around the bank, saying that given this distance here, I would expect most of these individuals to be guilty. Actually, a p-value is per gene. So this is just one test. And usually we say anything with a p-value below a certain point we think is associated with the biological condition or the crime. And we can draw these fences anywhere we want. We can draw them tight. We can draw them farther out. The farther out you get more guilty individuals, but at the same time you also get more false positives. So there's a trade-off here, and there's really no set rules to determine where you should draw these p-values. Usually what we do in bioinformatics when we're looking at lots and lots, say 50,000 different transcripts, is we'll use false discovery rates. And so as a p-value is important for per gene, a false discovery rate basically describes lists. So given within this boundary, I have a false discovery rate of 10%, that I expect not more than 10% of these genes, and they're very close, or these individuals, to not be guilty, to be a false positive. And sure enough, we have 90% of these are red. And that's usually what we do in statistics. We can also, which doesn't get utilized a lot, which is a great thing to do, is to see if more than one bank's robbed. And we can do this in bioinformatics. If you're investigating lung cancer, get 3 different lung cancer studies. If this gene or this individual was by these 3 banks when they were robbed, then the probability that they're guilty goes way up. So we can do lots of things, but again, these are arbitrary. These are strictly mathematics, and it doesn't really get us into how do we use the biology to kind of flesh this out. Okay, we're still staying with the same analogy. Uh, and I always ask this when I give this lecture is what other information would help you to decide who's guilty or not? And criminal history, obviously. Remember, you're the investigator. You have access to Scotland Yard. You know, if were these individuals guilty of a crime before, and if so, is it similar to the bank robbery we just that we're investigating? Now, just because somebody, and this goes in biology, because somebody has been guilty of a crime before doesn't mean they're guilty of this crime. And obviously, the reverse is true. Just because somebody has never been guilty of a crime doesn't mean they can't commit one right now. I think this, this kind of helps us isolate, who gives us a flavor, but it doesn't really tell us exactly which genes are important here. You can think of this as the usual suspects that, these people probably guilty of the crime, maybe not. We don't know. Here's an example of functions, and you can find this in, gene ontology gives these biological functions. And what we're doing is we're finding things that are overrepresented in our data set. This are some of the functions overrepresented in invasive bladder cancer. And you can see it makes sense. You've got all these things, all these genes here associated with cancer. The box represents the number of genes associated with it, the size does. Cellular movement going on makes sense with invasive cancers. We got your blood system involved, immune system, and a decrease in apoptosis. And again, this all makes sense, but are all of these genes critical to what we're looking at? And again, this gives you an idea of what you could potentially focus on, but at the same time doesn't give you a definitive target. So besides criminal history, what might be another important information? Well, I think, if these individuals were previously in a gang, that would be important. And we kind of do this with bioinformatics that, say I've got these 5 individuals here, and based on my database, they're known to be in the same gang, and there's only 7 people known to be in that gang, and 5 of them just happen to be walking by the bank when it was robbed. What is the probability that they're guilty? And obviously this goes up. That I wouldn't expect to see all these gang members by the bank just by random chance. And we do this with pathways too, and it's based on very simple mathematics actually. Well, again, these gangs can be very large. They can be small and powerful, and they can be doing very destructive things in your cell. But usually what you find is that these gangs aren't very well defined, that our knowledge is only as good as the intel we have. And this is all based on the scientific literature. And typically what happens is these gang members gangs share gang members, and they can work together. So it's very difficult to determine if there's truly one gang or is there a combination of both. And we'll get into that when we start talking about networks. So how does this work? Say you want to determine whether this pathway or this gang is playing a role in the particular biological condition you're investigating or are guilty of the crime. The statistics behind this is pretty simple in that, say this pathway has 40 members, or this gang has 40 members, and you want to determine whether this pathway is playing a role in, say, the 300 genes that you found that were either upregulated or downregulated in lung tumors. Well, these 40 members are part of the 21,000 total human genes that we know of. And that you can think of these would be 40 pink balls in a sea of 21,000 green balls. Given that these are the genes that I know are different in my biological condition, what are the odds if I just drew randomly 300 times from these 21,000 genes that would get 1 pink ball? Well, the odds aren't very, that would be fairly likely. I wouldn't say that this pathway is overrepresented in my genes. But now what if I get 3? the odds of that go down dramatically. Uh, Now I might say, yeah, this might be playing a role in my system. But now what if I get 7? This is highly unlikely. I would just draw 300 times and get 7 of these elements. In this case, I would say, yes, this pathway is probably playing a role in lung tumors. Um, and basically, we can do this. The statistics behind this is pretty simple. It's just basically a Fisher's exact test. That given a limited number of genes and a biological unit, I can tell in any list of genes, statistically, the probability of getting this. And we can do this for anything. We can do this for genes known to be affected by certain drugs or upstream regulators, networks, things associated with particular biological functions. Any contained biological unit, we can test whether it's overrepresented in any list of genes that we want. And this is the basis behind bioinformatics. As simple as it gets. Okay, so we talked about the pathways, the gangs, and again, our ability to figure out the gangs or the pathways is based on our intel. How much do we know about the system? Sometimes it doesn't work like that. That there's all kinds of these agreed to pathways have been devised by experts from across the world, and that in many different systems, and that what you see with these pathways are kind of idealized pathways. And just like real life, I wouldn't expect a gang in Detroit, the same gang in Detroit to be the same one in, say, Chicago. It's going to have different members. It might be doing similar functions, but it probably has different relationships than we think about. So in this case, another thing we might look for is just any relationship. So given here on these yellow, say these 3 all have a criminal history, but they have no known criminal association. But on a personal level, these 2 work at the same, or these 3 work at the same auto body shop, say, and this guy, this is the sister of this guy whose boyfriend is here as well. We've got a new network. We've seen maybe this is something we haven't seen before. That even though we don't have previous knowledge of them working together criminally, maybe that's exactly what they're doing. And we can do this using some of the software out there. Is there any relationship? They don't have to be in the same pathway, but is there anything out there that would say that they have some type of relationship? New gangs, something that we just aren't aware of. And that, and this happens a lot, that a pathway in a lung cell will be much different than one, say, in a nerve cell, or a muscle cell, or a brain cell. This is an example of that. For this instance, these were genes that were correlated to a gene called junction plakoglobin, or plakoglobin. What you can do is you can find genes that look like other genes across large amount of samples, and that's kind of what I did. I just correlated gene expression to see if I could find any genes that we didn't know about that might be interacting with junction plakoglobin to help it do its activity. Given here is the top network produced by these, I think it was about 150 genes that were correlated to junction plakoglobin over 166 lung cancer cell line cells. And as you can see, or as given here, the odds, so anything with an arrow, a solid arrow would be a direct relationship. Anything with a dashed arrow would be an indirect relationship. And the odds of getting all these biological relationships just by random chance, so again, this isn't pathways here, is 1 times 10 to the negative 65th. So there is, obviously, based on statistics, there's some kind of relationship. There's some signaling pathway that we weren't aware of. And if you look, the only genes that junction plakoglobin is known to interact with are those that are connected in the blue, based on the literature. What this allows us to do is now, hey, I think junction plakoglobin is acting with a bunch of different genes that we didn't know about. This is a new gang that we have for this gene in this particular cell type. And this is a powerful function that I use quite a bit. Okay, so we're still staying with the gang analogy. But what happens in this scenario? That say I have 5 individuals, they all have a criminal history. They are not known to be in the same gang, nor do they have any previous relationships with each other. But they are all known to take orders from this individual or gene here, the godfather, if you will. And we see this a lot, and especially in expression data, that the main players or the critical components in any system, you don't have time for them to be made from scratch. That if I wanted to get somewhere fast, I wouldn't build a car, I'd call a cab. And that's what your cell does, is that the important things in your that happen in your cell usually happen by phosphorylation or you remove an inhibitor. You need immediate action, and so we miss this on the transcript levels. But what this software allows us to do is that I might not see this individual, but I know he gives orders to all these people, and they were all around the bank. So even though he has an alibi, I know he's giving orders into my system. And if you think about in biology, that's really what we want, is that if you want to potentially cure a disease, you want to find these godfathers. You want to find these things that affect lots of things in your system. And the software allows us to do that. And let me explain. So this is the upstream regulator function of IPA, or ingenuity pathway analysis. So given here in the middle is this chemokine, interleukin 1A. And I never see it change. It's in your bloodstream, so that it's produced somewhere else. We never see this production, but it travels to the tissue and we see its effect. And we can tell, given the fact that, so in this particular example, interleukin 1A, even though I don't measure it directly, there were 43 known targets in my gene list. And again, this is the same Fisher's exact test. That probability of seeing all these targets attached to this particular molecule are 2 times 10 to the negative 17th. So even though I don't see the rock, I see the waves that it makes in the water, and I can follow those waves back and find that rock. And not only can we tell whether it's potentially active in our system, we can tell the direction of its activity. That given out of these 43, the vast majority of them that interleukin 1A is known to induce, which is shown by these orange lines, they go up, and this is given in red. You can see all the genes it's known to induce all going up. The 2 genes it's known to inhibit are going down, so green would be down in our system, and blue would indicate an inhibitory effect. The only ones that aren't following this activation state trend are these 4 here. Given the overwhelming evidence that interleukin 1A is more active, not only in our system, but more active, we can give it confidence scores. We can say we have the confidence that it's 5.5 times more active above just random noise. And this is a powerful tool. And not only can we do this, but once we get these godfathers, that's how they calculate the confidence score. Again, very simple math. And not only can we do this for interleukin 1A, but we can do it for microRNAs because we know the targets of those, even though we're not measuring for microRNAs. Chemicals. If you're researching a disease, you find that a bunch of the genes affected in your system are also affected by a chemical. You might want to use that in your system. And potentially drugs. So there's a lot we can, even though we can't measure these kind of master regulators, we can determine if they're there. And that's the kind of key. Then what we can do is we can take these causal networks. We can take these upstream regulators and start making flowcharts for them. And that's exactly what we do in biology, in that I don't want the low-level thug, selling drugs on the corner. I want the mastermind. I want the godfather. And, we kind of do that in biology as well, is that we can start taking these upstream regulators, and now we're making basically a skeleton to our expression experiment. And I would say that this, given here, even though we don't measure most of these, are probably the biggest effectors in any given system. And this is kind of the goal for bioinformatics. How do we get to this level? How do we take all of this information, use it to find these specific things that affect many different other aspects of whatever we're looking at. Where do I find array data? There's many places. Uh, Gene Expression Omnibus is probably the most common place that people deposit um, their array data. Array Express is the European version of that. You can have data sets that are specific to a particular disease like cancer, Oncomine. The Cancer Genome Atlas is an incredible place for lots and lots of cancer data. Where to find things to help you analyze the data. There's some open source systems. Broad Institute has a whole set of tools. I would say David is probably the most um, popular freeware solution out there. String, Cytoscape, which will help you visualize it, put in that kind of network type structure. And then you've got commercial software packages. I would say the most popular, the most well-used software is Ingenuity Pathway Analysis, but we also have GNOME out there, and I think there's also a GeneGo. I hope they're still in business, but. And where to find me? My email, website, Facebook. I have YouTube videos where obviously if you saw this, you found it there, and a Spotify playlist to help you have music for when you're doing this analysis. All right, thank you very much.